Okay. I don't want to waste time. I mean, as long as I meet you. Okay. Let's try and do the lecture. I hope you will be not too sad to hear that this is the last lecture in the series for. I hope that then, you know, loud crying starts happening. But anyway, that was asking, is this the last lecture? Indeed it is. So, it's number 14. We did this for 7 weeks. And tonight, or am I wrong informed? Yeah, I think tonight is the deadline, right? For your last HMM assignment. Yep. Okay, so then you're done. Well, I'll just do the exam on Tuesday. I think I have laid the last slide here, so that you all know what time you have to be where. Okay, so today I'll do a little bit. Hey, this looks distinctly different. Oh, nice. See, it's okay. I'm trying to go. Don't blame me. No. This is computers for you. Okay, so a bit bio, a few loose ends perhaps, wrapping up some of the principles, value mutation. We'll talk a bit about phylogenomics, 16S, how you can trace your ancestry. You know, if you didn't, if you're still thinking about nice Christmas presents, I'll have a piece of advice to you. We talk a bit about comparative genomics. What can you do? So, how could you use and how can you, utilize the concept of conservation? We've done that, of course, to some degree already. We'll do more. I'll talk a little bit because they're really interesting about telomeres, because they have an implication in cancer. It's one of the most serious diseases on this planet for mankind. Technologies to look into regulation and find all kinds of biological short sequences that act as motif. We've done, of course, the motif lectures. I'll talk about phylogenetic footprinting once again. I touched upon that in earlier lectures, but I'll do the pendant of that. It's phylogenetic shadowing. Then this is to give you a little bit of an insight in linkage disequilibrium. This was the old geneticist, but it still plays a role. And I'll talk about SNPs and haplotypes, single nucleotide polymorphisms, and derive from that haplotypes at the very end. And then we'll wrap up, and if we're lucky, there's a bit of time for questions. Are there any, let's ask that first, did you think about any sort of overall questions you'd like to ask? So we don't need to reserve half an hour for this. Okay. Now, okay, let's start quickly. I think I touched upon this already. Would you think that there's the highest life form on the planet? Some people claim that we are as humans. I don't know. But genomically, we're nothing special, right? We're just quite mediocre when it comes to the size of our genome. And here you see the numbers of genes, and so people said, look, oh, if we don't win in terms of sheer size, the length of the DNA, then of course, we are so more complex, so then we have more genes. It's not true. And there is hardly a relationship between the size of DNA and the number of genes. That is not as correlated as you would think. And this is a famous human, you might know the name of after this course. I hope you do. This is Mr. Darwin himself. All right, so here you see CMIs, and there are other even much greater genomes that are far greater than this. And here you see that length. Here you see, for example, amoeba. Lower life form, really quite a bit larger than we are. What is this? 200 times larger. So respect to these animals, right? So genomics teaches you a few lessons. So let's see what we can do here. So I started alluding to phylogenetic markers. What do you, if you want to find out, if you want to compare species, on what basis do you compare them? If you want to find out about the phylogeny, then you need markers as phylogenetic markers. And 16S RNA, and I say this so I can be short on this because that came earlier. The name Carl Bleuzy comes up here once more, and how it can be used. So in higher life forms, in eukaryotic cells, you go via the mitochondrial DNA. So you take basically bacterial DNA, and we have bacterial DNA as well, but in a mitochondria, right? It's the cell organelle that has a primordial origin, alpha-lactic proteobacteria, and so that you can use. Okay, so we alluded to what should a marker be, and I think even the last lecture I mentioned that a bit. But no, it should be conserved enough so you can align it, but it should be different enough that you can use it to find differences between species. And that is what 16S RNA is doing. The role of 16S RNA is currently becoming, with all of the sequencing activity, less and less, because you can do it based on complete genomes and or more genes. So that is happening now. So we are in a transition period, but of course this still has a large role to play. And in some clinics, still 16S is used a lot. Okay. And another thing is, lateral gene, horizontal transfer, you remember, and I'll have a few slides later on to fill you in on it. That is bad news for geneticists, because stuff is not exchanged through lineaging and through offspring, but 16S RNA genes suffer relatively little from that. Okay, and this is tree you see with the three kingdoms. Right? Eukaryotes here, Archaeobacteria, this interesting group that was discovered the latest, and the bacterial group here. Prokaryotes, this was all called prokaryotes. You had a subdivision in two, but ever since the Archaea, the name prokaryotes becomes problematic. Okay, horizontal gene transfer, standing in the elevator next to someone. You leave and you have sort of subsumed some of the genetic material. That's what bacteria can do, and they can do this in various ways. So this is called horizontal transfer, lateral, as opposed to vertical transfer, and that is genetical, so lineage via offspring. Okay, so the three main modes that bacteria can do this. They can build whole structures, so little tunnels to exchange material, and transduction, conjugation, and transformation. That is here, that you really build it in. And as you can see, a lot of these DNA structures in bacteria are circular. They're called plasmids. Many bacteria have quite a number of DNA copies, but sort of circular DNA. The circle has one advantage, and we'll get back to this during this lecture even. It doesn't have a beginning and an end. Right? Your shoelaces, what is always the problem? That the ends of the lace gets a bit crinkled, and you can't stick to the hole anymore. So you do special things. But more about that in a moment. So this is really, and early on people hoped that at least for bacteria, horizontal transfer doesn't play a big role, but the role is so big that we really have to keep track of it. And it's one of the main reasons, of course, of bacterial resistance against antibiotics. If one of your bacteria in your intestine gets resistant through horizontal transfer, quite a few others will have it, and that will then make this process even more dangerous in that sense of acquiring resistance against antibiotics. Did I tell you the clever thing that we tend to do in this country? We have more chicken in this country, for example, than the population, human population. So in hospitals there are 3 to 5, I'm not quite sure, antibiotics that are left on the shelf, left aside for when a real bad thing happens, right? Then we have at least this one, we hardly ever subscribe it, so then we can save these patients if they don't react well to other antibiotics anymore. In the chicken industry, they use those 5 routinely to fight any small disease. And that's the scare, that eating chicken is a problem if it's too raw. You get those bacteria in your body, a bunch of bacteria resistant against those 5 sort of SOS drugs, and that is a problem. So I think it's not clever that well-developed countries do things that are devastating in different communities. That do not bode well for one another. So data sciences we need to know. We need to know these things. It's hard to keep track of. That's a problem. Okay. How can you sort of find out about horizontal gene transfer? You compare to humans, and is there now some transfer? And very often you can say, look, these are bits of DNA if it comes in. Also, maybe at some bacteria, when this is another species, that that transfers a bit in it, and there are differences, general genomic differences. You have a chance to be able to tell the red part from the blue part. And that's sometimes important. And what can that be? That is that next slide. For example, compositional bias, if you want to be. And you find that a lot. Certainly if a bacterial gene gets in human, very often the GC content, so the composition of the letters is quite different, so that you can say, hey, at once here in the DNA, something strange starts happening. And then you could start guessing that is the result of horizontal transfer. Yeah, and you see that, that in bacteria, the composition, GC content varies wildly. Yeah. Lots of differences, and that is good, so then you can pinpoint the things. Yeah, here this is about how important it turns to be, but you can read that at home. And this you can read at home as well, just to fill you in a bit more. Okay. Tracing your ancestry. I thought, you know, I need to suggest a few Christmas presents, so they will come now. So you can give to your loved ones. So how can you trace? What is the problem? You know, that we are completely mixed, and for any self-respecting geneticist, they hate sex. Sex is a big problem in life. Okay, you need it perhaps every now and then, but to study the stuff, it's devastating because it mixes up everything. Now, where does it come from? After a few generations, you have no clue anymore. So that's really a big problem. So they hate it, right? Through biology, they hate sex. And but there is something, a way out, and that's great. One of those ways out is mitochondria. Why? In mitochondria, there's their own copies of DNA. They are of bacterial origin, I told you. What is the great thing about mitochondria? That's the fact that women add far more to life than men. Sorry to say, gentlemen, we enjoy it, but you do less. And already at the very beginning, because an egg cell has basically all of the cell organelles, like the mitochondria, and the sperm that is going to just insert a little bit of DNA. With one of the 23 chromosomes that's crumbling away, I will come in a moment, the Y chromosome, which is about the definition of masculinity. But anyway, because mitochondria are of maternal origin, that's fantastic. There is no sex around mitochondria, right? They go from mommy to the children and then on again. So there is no mixture, per se. And also what is nice, that there is quite a bit of evolution in mitochondrial DNA. So the mode of change is even larger than nuclear, than our normal DNA, right? So that's great, because if you wait a while, really stuff is happening, but at least you know when we're here, you know, it goes on. There is no mixing. So you can pinpoint, and if you want to say, you know, people in regions, the mitochondrial DNA will stay more the same than others. That's great for a nice Christmas present, which will come up in a moment. So it's maternally transferred, and that's great. So you can use that nicely. So another thing is, in the same way, the Y chromosome. Why is that? Because you know that ladies get acquire two copies of X. They are far more gene-rich X chromosome than the Y chromosome. Even there have been some estimates that the Y chromosome is falling apart, it's deteriorating, and some, maybe worrisome, I don't know, that in 2.5 centuries from now, 250 years in the future, it might be that it's so crumbled away that maybe males stop existing. So then we need something else. Then, no sex please, because we're human, nobody's no sex because we're British. So maybe that a few men will kept in special conditions that the Y chromosome is just okay. I don't know what society will get, but something like that I reckon. So we go to maternal societies, I guess. But anyway, we're not, you won't be there in 2.5 centuries, I certainly won't be there. So for us it's not a real problem, you might say. But, so, and the nice thing about the Y chromosome, at least, males have an X copy and 1 copy of Y. And that comes from father to son to son. So the same way, paternally that is transferred through the generations. There is a little bit of a problem, you might say. What do you do as a woman if you want a nice Christmas present, able to trace your ancestry? You don't have a Y chromosome. Wow, okay. At least you had a father, that's pretty clear. If you find a guy, at least you can use that. So you need to either take a brother or your father, and then you have a chance of getting somewhere. With your brother, there is a degree of uncertainty. You need the same father, right? With father, do mitochondrial, I think it's safer probably. Anyway, because there, when genomics came and DNA testing, that was a little bit shocking. You know, the number of illegitimate children was estimated at a certain level, and that was kind of a surprise, of course. It was more than people thought sometimes. Anyway, we're not on about that at the moment. So, why is another thing? So, you know, about precision. So, you know, of course, you know, it's about markers. These tests, they do not sequence the whole thing. They have some markers. They do small regions, and you know, of course, the more markers you have, the more points you take. What is happening there, the better you can pinpoint, of course, exactly where you are. And this slide is on to that. So you can, at only 80 markers, you can go back fairly precisely in 27 generations. How long is that? Generation, 20 years? No. It's already a couple of 100 years, not too bad. So you can go back to quite a bit already. Mitochondrial is more precise, but you need a few markers. But at least this has a power as well. So now it comes to the present. How's €50? How is $50 for the present? Is it? I'm sure. 23andMe, the company. Buy the kit. It's now for sale, right? It was 80. It's now 50 for Christmas. See? There you go. You send it, and this lady goes on. You know, she turns out she's done it, and she was Northern, Western, Southern European, and she's partly Chinese and a bit of Arabic and a bit of Asian. If Chinese is Asian. So you find out. One of the things they also know that 4 to 17% in us is Neanderthal. You know, Neanderthal, you think, oh, they were stupid people. Now it turns out, no, they're not stupid. They were even cleverer than us at the time, right? So you don't need to be, you know, you can be proud of that. It's not a problem. And, but you know, so we mix a lot. So even Homo sapiens has a lot of other origins. Even at that level, they can find things. So genomics has enormous power. Oh, here it is, add to cart 79, but here you see 50 off. Hold on. Up to 50. Oh, this way. See, all these guys are marketeers to bloody hell. Up to 50 off. There is nothing off. It's up to 50. Anyway, 79. Okay, a bit more expensive than I thought. Then you can go to the competition. MyHeritage, they do the same thing. They claim they have the biggest database. They have all the regional DNA, and they map, you know, a lot of markers. They map it onto all those regions, and then come with a nice report on, you know, where you're from, right? And to a lot of people, this is quite a surprise. And I can tell you, these tests are not popular with Ku Klux Klan people and so on. They get really some bad news all the time. You thought you were a white guy? You know, that sort of stuff. You know, it's great. And racial, I think I told you this already, yeah? If that's a great power of genomics. You know the out of Africa theory. The idea is that we, you know, walked once or one while ago out of Africa, and through the Balkan, you know, the mankind entered Europe and then separated over Europe. So that means that still you find in the black population, what is the black population? The African population, the variance is much larger. So if you have African people, sorry, they're sort of everywhere here. Big back, oops, sorry again, to the other side, yeah? There's a nice Clouseau pink pencil, you know that one? Okay, anyway, so if we have the white population, that is roughly here, say. That means if you talk about differences, Ku Klux Klan stories would say, you know, what we want to see is, you know, this is black and this is white, right? The enormous divide, and this is so much better. That's the idea, right? And now this is the situation. So what do? You say now, that means that a white person is more like a black person than a black person is like a black person. That's a story that Ku Klux Klan finds hard to defend, right? That's, you know, that's not what they would have liked. So they hate genomicists and so on. And it's all fake news, of course. And, you know all of these stories that go around these days. Anyway, Understanding, still all about understanding genomes. Here you go. I'll fill you in a bit on a study that was done in the Broad Institute. They did sort of simple tests, but they're quite revealing at the time, and still lessons good to bear in mind, of 4 yeast species. And what was conserved there? Okay, so they looked into real genes and regulatory elements as well. And they, okay, stuff came out, but I'll get to that when we get there. Now, wartime story, Second World War, England. They were bombing Germany at the time. They were winning, they thought. But what happened is, some planes, after they went over Germany, of course coming back, refill, go back again, but some planes were shot down, they didn't come back. Then the army thought, you know what, we should do some really design and data science, right? We should look at the planes when they come back, learn from the patterning of bullet holes, learn from it, and build better planes, so that, you know, more come back and we can reuse them. That was the idea. And they hired Abraham Walt, a mathematician, to sort out how to do this. And I'm talking you into, you know, evolution and so on, right? So you can sense what is DNA. Anyway, so this is a typical plane that came back. So the military, they said, oh, it's great, you know, you see how they come back? And you know what we should do? You see all these holes here, that's bad news, right? We have to reinforce these parts here, the wings. And here, you know, put plates there. I see already some moves that I like seeing. That was the idea. And then Abraham Walt, being a mathematician, well, you can tell it. You don't need to be a mathematician, right? The thinking is flawed. What is not okay here? Because what is this? What was this? This was a successful plane, you might say. It came back. So what do you think is behind? So what should you focus upon? Not on the bullets, the holes that were attracted. Just what you probably didn't see. And if you think, where's the pilot, for example? He's not here, I can tell you. Not here, he's there, right? Maybe the cockpit says here. Anyway, or maybe he's here somewhere in the cockpit. What is here in the plane? Well, you have to know the plane, but the engine is here. So what is the story? What comes back? This is not a problem. The lethal mutations are not these. These are mutations that the system can handle. The lethal mutations are probably there. So from this study, you should take, that's great, we will not reinforce these bits, we'll reinforce the bits in between. That was the real story, right? And that is what Abraham, how is it called? Abraham Walt advised them, and they started doing it, and indeed it worked. You know, the planes were really stronger when they made the underside of the plane in these regions. They made them stronger with all kinds of metal plates. So, where have we got here? This is now with Darwin. This is almost phylogenetic footprinting. You try and find evidence to see what is important. And what is important is conserved in evolution. So, you know, of course, DNA here, the genome is the airplane. Mutations are bullet holes in this story, right? And natural selection is combat. Yeah? So this is an evolution. So we have functional regions. You better leave those alone. Mutations there will be lethal, but in other regions, intergenic or something, maybe it's not such a problem when you track mutations there. And that's what you exactly see in this example, right? Okay, on to the real study then. 4 genomes we'll look into. And so this is I think Jacques Monod, one of the important early scientists in evolution. The idea is evolution, you do not reinvent the wheel all the time. You tamper with what is there, you build on, right? Evolution has searched a tremendous lot, but I gave you one example that maybe you know didn't search everywhere. Immunoglobulin proteins, they're in our blood, they're very important for our resistance and so on against foreign invaders. These are proteins that are typically 500, 600 amino acids or larger. In industry now, they have developed all kinds of antibodies and stuff with 70, 80 amino acids. So the question could be, why is evolution be so stupid? Why make a very big protein with all of all the problems you get when you can do it small? The thing is, it didn't look there. Evolution looks, it's neighborhood stability. You have something, you start tinkering with it. You shoot bullets in it, you do little things, right? But you do not, you're searching in here in the space. And if you search long enough, you'll drift away and you'll get new species and everything you can see on this planet. But you do not, per se, there is no power to at once say, okay, let's go here and now look. Right? So it's neighborhood stability, that's how the traditions go. it searches a hell of a lot, and you already know that the PAM 250 matrix, how many substitutions on average is that per site? Remember? 2.5. Every letter has changed 2.5 times. I think that's quite a bit. And some more than others, so some really everything has been tried perhaps. So, but this is the base message, right? It's tinkering, it's changing what is there, and then see what happens. Okay, for yeast species, here they are. Saccharomyces is the word here, so that's the S here. And this is the famous one, yeah, right? We eat it every day, I guess, baker's yeast. Okay, so the divergence was, well, we don't know, but let's say somewhere in between 5 and 20 million years ago. And the nice thing is these genomes again are divergent enough to see nice differences and related enough to be able to compare them and find and align them and find the orthologous genes. It's only because you need to pinpoint, oh, this is corresponding to that in the other gene, right? Here is your paper. One of the young people is still young, by the way. It's Manolis Kellis, who's a successful young Greek by origin scientist at the Broad Institute in Boston. So the first step what these guys needed to do is lining up these 4 genomes, pinpoint the genes, and this is what they did. And you see there was a fair deal of conservation at the gene level. This, by the way, is also referred to as synteny, gene order. And here you can see that there is quite a bit of conserved gene order, right? In these 4 genomes, and that is good because then you are all the more certain that you still really have orthologous genes here you can compare that are supposed to do the same thing in each of those organisms. But you see that there are, every now and then there is a bit of blue here, and that is just local variations that stuff happens, yeah? Affecting synteny, and these might be some strange multiple matches you can have here, sort of repeat. But here this looks like an inversion of something else, or I don't know, inversion in the mapping, but I have to look into it. But these are sort of the exceptions, but you see most is bad. So, first bit of statistics they gathered. See if you would agree with this. So they compared all these pairwise, they compared them, the 2, and then they find, so all the time baker's yeast was compared pairwise with the other 3. And this is what happened. So some are further than others. These are the similarity in the coding regions, where they say the protein coding genes are. You see this is the nearest neighbor. So you see this. Do you see this? Does that make sense to you? The similarity is twice less, from 90 to 80, 10% different, 20% different, 15% different, 30% different, 20% different, 40% different. And in antigenic group. Why is that? There is more selection, of course, on this gene region. Does it make sense? Okay, next bit of statistics then. If you, they now looked for conservation across the 4 species, not in pairs, but across the 4, and that's why the identities go down, because now in the coding regions you go down. If you only count them when they're all 4 conserved, you see that now the identity level is at 60% now. And there are, you see here a few mutations. Here's another type mutation that is called the frame shift mutation. You know, remember what that was? There is something happening to it that either that you affect the multiple 3 situation, deletion of 1 or 2 or a multiple of that, so that you get another interpretation or another translation, for that matter, of your RNA material in proteins. And you see there's a lot of selection against it. And you see here that an intergenic region, of course, again, identity levels are down. Gaps are quite a bit more, so gaps are very much selected against in coding regions. Deletions, insertions are dangerous. And you see that frame shift is the most selected against, because that will, you know, obscure, will clobber everything that follows, right? You get a completely different protein, and very often it's a nonsense mutation or a truncation mutation that, you know, your protein is shortened, and that is normally not good for its function. So here you see the ratio of what is allowed, the twice we saw already. For gap it's 10 times more in intergenic regions, and for frame shift you see it even 75 times more in intergenic regions. Okay? So this all leads to the idea of these guys said, okay, we use this simple stuff, simple statistics that seem to work well in these 4 yeast species. Can we start predicting genes or seeing what happens? A lot is known, also at the time of the paper, 2001 I think it is. So could they refine the knowledge body that was there at that time. And so they started thinking, can we find a dummy method, a sort of very simple method to find open reading frames, likely gene regions? And they thought, because they say this is a high quality data set to compare stuff, so what is the advantage of doing comparative genomics? Can we really use conservation to make methods more sensitive and still have dummy methods that are simple, but maybe they still will work because of the divergence observed within across those 4 species. Now, this is what I already said to you. A gene is special in a sense that the protein can be 500 long, so the gene is 1500 long at least, if there are no indels. And all that time, there should not be a stop codon, right? All the way. Stop it. So, what's the frequency of stop codons? It's 3 stop codons in 64, say 1 in 20. Randomly, 1 in 20 positions is supposed to be stop codon. That means if you've got a long gene, there is enormous selection against the occurrence of stop codons, right? So maybe you can use that. And then what happens if you use this and also use the notion of conservation? And that is where this plot is about. I need to read it again. So red is where things go wrong. Red bars indicate frame shifts. Okay, so these are frame shift mutations. And frame shift mutations will, of course, quickly generate, if you like, stop codons. And you don't have stop codons for a while, but then if you do frame shift, you will find 1 in 20 is stop codon because it randomizes what is following, and you will see it. So here, and these was a known sort of gene, and they easily could write it off based on the conservation of these mutations they saw across the 4 species. And this example clearly, in terms of the stop codons you would get based on this, that by means of conservation, they could write off this gene in baker's yeast, because all these genes were known in baker's yeast. It's the most widely studied yeast species still on the planet. So they did these type of simple things and then tried to find genes and so on. So the results were from many genes. They said the studies were done with by high confidence. They could write off 15 out of 4,000 known genes. But the total number of a bit over 6,000 they refined down to say 5,500. By this, they said these open reading frames probably, when we look at the other species and look at the conservation signals, are not real genes. And the idea is that these genes that are there will play a role in each of those species because they're similar enough. And so they could redefine the start and the stop codons based on conservation. You have an ATG, but it's not conserved. Here further down there is an ATG that is conserved across the 4. That is probably the start codon for the species. And they could correct, if you like, 5% of the genes in that. And they found, by doing this, a whole couple of new introns as well. Okay, so another thing you can do using conservation is finding regulatory elements. You've mentioned this. This is, remember how this is called when you specify something like this? A regex, right? Regular expression. So this says CGA, and then I don't care for 5 positions, but then I want to see TGG, and that could be some regulatory element, right? So they did a study. They had over species where some of regulatory elements were experimentally known, and they started looking what they could do. And the idea is, what is a biological? How can you find out if this is a short element? How many times do you think this motif will occur in our DNA? 3.3 billion letters. Zillions of times, maybe a million times or more. How many of those will really be a biological regulatory element? Just a small subset of that number. So how can you tell them apart? What is functional, what not? And then again, you say, if you, in this case, the 4 yeast species, if you find conservation of them, then your guess will be, if it really has a degree of conservation, then you say this is a functional one, and other ones will be spurious ones that probably will not have a biological function attached to it. Okay, so here is what they started doing, and to find out how can you really get a handle on this, they started thinking we should use a random motif. So here you go, it's counting how many, and this is a specified motif, and I think the Gal4 motif was the motif on the preceding slide. So they find, does this make sense to you? If you really look at one of the possible regulatory motifs, you find that this transcription factor binding site, because basically that's what it is more conserved, if you can see, in intergenic regions as opposed to the coding regions. So it is possible that they're there, but you know, normally upstream is happening most, but outside the real gene, in the regulatory section of genes, you will find them more. So in coding, you see it's low. Then they said, you know what, let's check this randomness ID. We'll take the GAL4 motif, but we'll randomize it. We'll shuffle the motif, and then look, and then do the same statistics. That is then what you see if you randomize such motif, and then you see that it's quite different. Even more of that happens in the coding region. That means, now, if you compare these two, you could say, as opposed to random, you find that real regulatory elements happen about 11-fold more in intergenic region as compared to the random situation. So you can do this because you, again, comparative genomics, you use it across about the conservation over 4 well-chosen species here, genomes. Okay, so, and this is what you should know by now. These pictures have come. This is the situation with regulatory elements and transcription factor binding sites in prokaryotes, in bacterial. This is in eukaryote, more complicated, and you have all kinds of switching networks in eukaryotic species. This is the situation in a blood stem cell, for example. And here you see, if that picture has reached you already, that you have, you know, I think I showed this to you earlier on. Do you remember? By the course, maybe. Anyway, do it quickly again. These are 2 genes. You have the two, of course, the plus strand and the minus strand of the DNA. So you find motifs in 2 directions, you might say. This is the gene in the one direction, here the gene in the other. This is the intergenic region, and you see that you get all kinds of, here are these GAL4 motifs, and the point here is there are multiple positions where this transcription factor that binds this motif could bind. This one will be blocked for whatever reason it might do here. Incidentally, this bit of research clobbered another certainty. This was the GAL4 motif. They look at these 4 genes, have it experimentally verified. These biologists come back and say, right, here we found another GAL4 motif. They looked here and they said, bloody hell, it's not CGG, it's something else. So here you go. This bit can be more varying than that one here, right? So you have sometimes, if you have experimental evidence, you have to retune what you think you know. And here are a few other transcription factor binding sites. And here you've got the famous Tata box in 2 directions. This is a point of attachment for the polymerase to do the copying, right? If this is not there, you're in deep trouble. The gene will not be transcribed. Okay? So this was a sort of a simple piece of research they did like that. And by doing that simple conservation again, they found all kinds of new regulatory motifs, and I think the majority in the meantime have been confirmed. So the power of comparison, what is conserved, that has a larger chance of being biologically relevant. And so overall in the study, this is what they could do. They had an exact count of that number of genes. They had better gene boundaries, new introns, new motifs. They could sort of count better what is really the coding part of the genome. And that turns around. So in us it's 2%, it's 70% in yeast, and they could find repetition. So simple comparative studies can tell you a lot. And here you get a bit more detail on this 5,500 genes that were reconfirmed or corrected by means of this fairly simple study. Okay? I'll do this one thing, then we go for coffee. This is sort of nice. This is about, I showed you the DNA in a bacteria. So this is about, in fact, telomeric regions. Telomeres are the ends. We have linear chromosomes, linear DNA. They have a beginning and an end, 5 prime, 3 prime, it's called. And that started off from bacteria they were around. So at one point in time, these bits were broken. And this is just to show you, it doesn't matter the details so much. This is if you see something, these are the genomes in yeast. Is it yeast? I think so. Yeah, it's the same study. So things happening, and basically what you can see here is that a lot is all the time happening at the beginning of the end. You see that? All kinds of variations are happening there. Sometimes stuff in the middle happens, but you see, for all of these chromosomes, you see stuff happening at the beginning. And if you zoom, sorry, I thought I had to zoom in. It's not here. It's not here. Doesn't matter. Okay, so here it explains what you really see, but that's the point you should take home from this picture. So a lot of evolution happens at the ends, beginning and end of DNA. And why would that be? Now it's the picture to zoom in here. So you can see here, in these examples, zoom in, that a lot really is happening. So a lot of evolution is clustered in the telomeric regions. Does anybody an idea if there's any functionality about these ends of DNA? Do you know? Then I tell you. Telomeres, here you go. Telomeres are sort of, they're ends of DNA, but you will find that there is a lot of stuff around it. If you have these laces of your shoes, what do you typically find at the end of the lace nowadays? Plastic caps. Why is that? To not get in this annoying situation that you cannot fold it through a hole anymore, right? That is what DNA has to do as well. And why is that? But it came into play the moment, maybe the circularity was broken and we got linear DNAs. These were a bit unstable, these ends, and they started, what can happen then? More easily your DNA will resolve, will crumble at the ends. And if that goes too much, of course, that will affect your genes, and that is bad news. And basically, that has evolved in a ticking clock. Cells know how old they are by means of the situation in the telomeric regions. Because what is happening, these telomeric regions still tend to shorten over time. And the cell can sense how much it shortens. So the cell knows after each division, part of that region is gone, and typical cells in between 40 and 50 cell divisions, a cell will naturally say, I'm an old guy, I should stop. I've accumulated probably so much error over all the division. I should stop doing it because my fidelity is low. Younger cells should start doing it. Right? So cells stop and they might even kill themselves. And that's called apoptosis, right? Programmed cell death. And a large part of that program comes from the telomeric regions. Yeah? And here a few, sure you can read it home, but okay, so the split happened between bacteria and the eukaryotes roughly 2 billion years ago, and so then we became, and the problems in the shoelaces. Okay, so without proper telomeric structure, we all die. Species cannot live. So telomeric DNA is composed typically across all lineages with sort of shorter, very much repeating fragments. Vertebrates have this typical TTA-GGG, and then you get TTA-GGG, TTA-GGG, roughly 150,000 times easily that repeat, so it's quite a stretch, telomeric region. And in bacteria, this is, I will show you that thing later on, it's different, but then this is repeated, right? So, and what happens is, so you have this complex, these bits of DNA, there are all kinds of proteins binding it, but at each replication, roughly, they don't know, but depending on the species, 50 to 500 of those repeats in eukaryotes, for example, are lost. So the DNA is shortened by so much. 500 times 6 nucleotides. What was that? 3,000 gone. Boom, one division. Next division, another 3,000 off. So the cell seems to be able to sense that. And so this leads to programmed to cell death in the end if it happens too much. Now, then you see tumor coming in. There is an enzyme called telomerase that is capable of elongating, repairing, if you like, the number of repeats. It will put them back in. And that's great. This is the secret of a long lifespan, right? It's fantastic. The problem again is, the cells have this natural program like, I'm not good anymore, I shouldn't divide. These cells go on dividing. Very often these turn out to become cancerous. Yeah?
In the third bullet, what does basis bound by a large number of proteins mean? What do these proteins have to do with things?
So that is that plastic cap idea, that it should be protected. to be programmed. This tends to quickly become brittle and fall off, right? So that should be protected, so that you can sort of, on each division, these protein complexes are busy and then it's short, right? But that should be a controlled process. So you have all kinds of proteins. Yes, yes, all kinds of proteins, and I'll show you a picture in a moment. Right? Sorry, where you can see it. Oh, sorry. So it was already. So that's the point. Cancer, they fix. This is a natural process. Shorten, shorten, shorten. Stop dividing. Kill yourself. Cancer sort of resets this program and makes that cells can divide on into eternity, and that is bad picture. So a lot of medication in cancer, focus in cancer research is on these telomeres and trying to can we do something against this telomerase in cancer. That doesn't work anymore. If you do this too well, you don't want that each cell after 2 divisions so says, oh, now I'm an old guy. There's a balance in that, right? You don't want eternal life because that leads to cancer. But we all want a long life, so there is this. What do we do? So you might say cancer is a disease that makes you young, but in a very uneasy way, you might say, right? But okay, so they found some things, and there is now in big pharma focus on that as well, and that is something that binds, and here you see it already mentioned. They found that this G-quarter DNA, that's a bit of DNA that is GGG, and incidentally you see that there's quite a bit of GG here. What that can do is, if you have a lot of G there, it binds to it. This is a way of sort of a glue to bind of 2 DNA molecules, for example, the telomeric regions together. It is taken by this sort of glue stuff. And when that happens, the telomerase cannot elongate these bits anymore. So you sort of stick DNA together, and then it cannot be, cancer, this cancer protein cannot do its job anymore. So this helps against cancer. Of course, they need to find out, just tying stuff together, is that helpful for other processes or not? Is that a problem? So there's a lot of research here. And here you see that, these are these typical molecules here. And here you see, well, here's a blob of DNA, and you see, oh, this is a big protein that is there that binds to these telomeric regions. Yeah? Yeah. So, and it's specific domains do this, you can see that here. And here you see, well, there's gray, dark gray and light gray, where there's G-quartet, where 2 DNA strands are bound together based on these four genes, you can see there. So this is now, really an active focus in cancer research. Can we fight cancer by, not allowing that repair process, if you like. So that the telomeric region shortened by at each division. Okay, so that about the interesting region of DNA, the telomeres. Okay, a bit of now we drink coffee, I think. Right? The last lecture, I'm going to bribe you. Who wants coffee? Get away with it one more time, I think.
I thought it was 3. But I don't know where.
Yeah. No, I just looked at it and I thought it was the same as. Yeah,
so you're already doing it overall synthesis, but each iteration is just running the whole algorithm all over again, just with a different prior matrix.